Welcome all. My name is Saroja and I am an associate professor in the Department of Pharmacology. And today let us discuss about the anti-cancer drugs or the chemotherapy of cancer or chemotherapy of malignancy. We can call it by either names. And coming to definition of anti-cancer drugs, these are the drugs used for the treatment of cancer condition. Drugs used for the treatment of cancer condition are called as anti-cancer drugs. So what do you mean by cancer? So cancer is a condition that is characterized by uncontrolled proliferation of cells, and it is also characterized by the process called as angiogenesis. So if there is uncontrolled proliferation of somatic cells or bodily cells, it is called as cancer condition. And to supply nutrients and oxygen supply to the growing tumor, it requires a blood supply. So blood supply to the cancer cells occurs through angiogenesis process, that is production of new blood vessels to supply the nutrients and oxygen to the tumors or neoplasm. Tumor is also called as neoplasm. So this is about the cancer cell characteristics. So cancer occurs because of uncontrolled proliferation of somatic cells. Somatic cells are also called as body cells. Coming to somatic cell division. So let us have a good idea regarding the somatic cell division, which is very important to understand the mechanism of action of various anti-cancer drugs. So cell division in human beings. Cell division in human beings is of two kinds mainly. One is the somatic cell division, and another is reproductive cell division. So somatic cell division is the cell division which is characterized by the division of body cells, and somatic cell division, it involves the mitosis process and cytokinesis. And reproductive cell division, it involves meiosis and cytokinesis. Somatic cell division, it leads to formation of body cells, and reproductive cell division, it leads to formation of sperm or ova. And sperm or ova are gametic cells. Coming to somatic cell division. So this somatic cell division, it involves the interphase and mitosis followed by cytokinesis. So coming to interphase. So interphase of somatic cell division, it involves G0 phase, G1 phase, and it includes S phase and finally G2 phase. Coming to Z0 phase. Z0 phase is the phase in which the cell will be present in the resting condition. That is Z0 phase. And G1 phase is the phase in which the cell will be metabolically very active. In G1 phase, and it is also characterized by replication of centrosomes. So replication of centrosomes along with cell growth, all these events takes place in the G1 phase. So cell will be very active metabolically in G1 phase, and it is also characterized by replication of centrosomes along with active cell growth and cell proliferation. So all these takes place in the G1 phase. Next phase of the interphase is S phase. So S phase is also called as synthesis phase. And G1 phase is called as pre-synthetic phase. And S phase is called as synthetic phase. So why this S phase is called as synthetic phase? So because it is called as synthetic phase because in this synthetic phase or S phase, there is synthesis of DNA or there is replication or formation of new DNA takes place in the S phase. That is why it is called as synthetic phase or S phase. So in this S phase there is formation of new DNA, and all the components that are required for the formation of new DNA will be formed in the G1 phase. All the components that are required for the formation of new DNA through replication will be formed in the G1 phase of the interphase. Next, after S phase, there is post-synthetic phase or G2 phase. In this G2 phase, there is still there is cell growth taking place. Still cell growth is continuous. Still cell growth is continuous in nature. And G2 phase ends with completion of replication of chromosomes. So replication of chromosomes gets completed by the end of the G2 phase. So this is the gist of the interphases. So G0 phase, G1 phase, S phase and G2 phase. G0 phase is the phase in which there is resting condition of the cell, and G1 phase is characterized by active cell. Cell will be very active metabolically in G1 phase, and there is replication of centrosomes along with cell growth, and this phase, G1 phase, is called as pre-synthetic phase. And S phase is the phase which is also called as synthetic phase. So why this is called as synthetic phase? So it is called as synthetic phase because there is formation of new DNA by means of replication of DNA. So that is why it is called as S phase or synthetic phase. Next phase is G2 phase. So this is the last phase of the interphase. So G2 phase is characterized by continuous cell growth taking place, and along with the continuous cell growth there is formation of organelles, cell organelles, and proteins and enzymes that are required for cell division will be formed in the G2 phase. And also it is characterized by completion of replication of and it is also characterized by replication of centrosomes. So all of these events takes place in the interphase. Next, coming to mitosis. So mitosis, cell division. So mitosis, cell division, it consists of 4 subphases. So these are called as prophase, metaphase, prophase, metaphase, anaphase, prophase, metaphase, anaphase, and telophase. So these are the 4 phases of mitosis. Mitosis consists of 4 subphases. One is prophase, another is metaphase, next anaphase, and finally telophase. And mitosis is ends with mitosis. It is followed by cytokinesis process. Mitosis followed by cytokinesis process. Mitosis ends with the telophase, and mitosis stage is followed by the cytokinesis or division of cytoplasm. Coming to different phases of the mitosis. Let us discuss about different phases of mitosis. So first one is prophase. So prophase is the phase of mitosis which is characterized by the formation of paired centrosomes. So initially, before undergoing cell division, cell will appear in this manner. So cell, it consists of paired centrosomes. Paired centrosomes are present. So there is formation of paired centrosomes before the occurrence of replication, before occurrence of cell division. So there is presence of paired centrosomes along with presence of nucleus, and inside the nucleus there is presence of nucleolus, and inside the nucleolus there is presence of genetic material. So are chromosomes. So chromosomes will be present, or genetic material will be present in the form of chromatin. So this is the chromatin, and this is the cell membrane or plasma membrane. And these are paired centrosomes. And this is nucleolus. This is nuclear envelope. So before prophase, cell will appear in this manner, and once the cell undergoes prophase, cell appears in this manner, and here there is formation of paired centrosomes. So there is formation of paired centrosomes. Along with that, there is formation of spindle fibers. Spindle fibers will also get formed. So there is formation of spindle fibers. So there is formation of spindle fibers. So here the spindle fibers are formed. Here the spindle fibers are formed between the two centrosomes. These are the centrosomes. Spindle fibers are formed between the centrosomes, and this is the nucleus. So in the nucleus, so chromatin, it gets condensed. Chromatin gets condensed and it appears in this way. Chromatin undergoes condensation and it appears in this manner. And here there is centromeres. And this is formation of early mitotic spindle. This is formation of early mitotic spindle. So early mitotic spindle is the characteristic feature of prophase. Early mitotic spindle is the characteristic of prophase of mitosis. And next, so in this prophase, there is disappearance of nuclear envelope and nucleolus. So here, this is the nuclear envelope. So by the end of the prophase or before metaphase, or it is also called as prometaphase. Prophase is also called as prometaphase. So or by the end of the prophase, there is disappearance of the nuclear envelope and nucleolus. So both of them will be disappeared. So this is also one of the characteristic feature of the prophase. And early mitotic spindle is formed. And chromosomes consists of two sister chromatids. In the prophase or prometaphase condition. Next,